Hello, welcome to this next installment in our Optics for the Cloud lecture series, public lecture series. If there are any here from outside Microsoft, from the university or elsewhere in Cambridge or further afield, thank you for coming. In this particular lecture series, we've got Rudolf Oldenburg coming to speak to us. So Rudolf is a senior scientist at the Marine Biological Laboratory in Woods Hole, Massachusetts, where he develops light microscopy techniques for studying living cells. He's got an interesting background. He started out in Munich, Germany, and then a Max Planck Fellowship to work in Grenoble, France, and then where he received a PhD, and then back in Germany, and then finally to the United States, first for postdoctoral work at Brandeis University, and then more recently at the Marine Biological Laboratory to work with Shinya Inoue. Is that correct?
That's
pronunciation?
Right. Yes.
Okay, great. Starting a really interesting and fruitful collaboration working on techniques for orientation and polarized light microscopy. So Oldenburg is the inventor of the LC-PolScope, an instrument that enables biological discoveries and has become an indispensable tool in clinical and research laboratories around the world. In his current work, he works on the third dimension of orientation imaging while continuing the tradition of cutting-edge research through interdisciplinary collaborations, combining technical, physical, and biological insights to find the answer to life's pertinent question, what is life? I have to say, from my point of view, it's great to have Rudolf here. The interesting thing, of course, that he shares with many in the audience today is this interest in birefringence, which we're working on in one of our projects here. And I have to say, as a computer scientist, seeing many in the team join us as optical physicists of various sorts, the name Oldenburg has always been spoken over the last several years, always with tinges of somewhere between admiration and awe, and perhaps a little bit of reverence. So it's wonderful to have you here. And we very much look forward to hearing what you have to say to us. Thank you.
Thank you, Richard, and thank you, Ariel, for inviting me here. It's a pleasure to come from Woods Hole. I'll show you in a moment where that actually is. But this is one of my heroes here, the mantis shrimp, because it has such a sophisticated optical, an eye and an optical system that it can detect different types of polarization and has 12 different types of photoreceptors. So you can see from the UV to the infrared. Let's see here. In fact, many animals can detect polarized light, but not the human eye. So we don't really have an intuitive understanding of what is polarization, even though it is a very basic property of light. It's in fact the Maxwell equations. Maybe some of you have had contact with them. They only describe polarized light, and unpolarized light is kind of a statistical property of light. So if there were an audience of cuttlefish, would be no problem to talk about polarization, but the human eye does not have. So I come from the Marine Biological Lab. It's let's see, it's here is Boston, and this is Cape Cod, and here in this location is the Marine Biological Lab. It's 1888 it was founded and was an independent research lab funded mostly through grants from from agencies and philanthropy, and then it became kind of we have recently this were recently affiliated with the University of Chicago, and it's really a very nice collaborative setup now. And for us who are interested in imaging, and in particular, as you all know, computers are everywhere now, and in imaging they become more and more of a tool for analysis, for generating images, and so have a very good group, the Chicago in medical imaging, which uses, for example, computer tomography and MRI, where the original data don't look like an image at all, and you have to kind of generate the image or the 3D object through an inverse, by solving the inverse problem. And that's really something that is in regular imaging still a hurdle. While it is easy to generate an image from a known object, if you know its physics, going from the object and an imaging system, it's easy to generate the image from it. It's physics. But going the other way, from the image inferring the object, is still an ill-posed problem. Mathematically, it has many solutions often, and it has many solutions that are not physically meaningful. And so that's a problem that we also try to solve in conjunction with polarization, which is polarization typically introduces a nonlinear relationship between the image and the objects, and that so it's even more complex in a way. Let's see. So the collaboration with Chicago is really very helpful for us, which has a strong medical imaging group. And this is what MBL looks like. These are all parts of MBL. Woods Hole is really a little research village, in that the Woods Hole Oceanographic Institute, maybe some of you have heard that, is there. They have a focus on oceanography, on physical oceanography, chemical, and so on. They have these deep diving vesicles that discovered the Titanic, and so that's their claim to fame. And then there are many other, the Woods Hole Research Center, which is policy, in part policy and environmental policy. And so there's a fair number of colleagues in different areas in Woods Hole. Let's see. So unfortunately, I have to, the demos didn't quite work out. But I will tell you about this LC-PolScope technology that we developed. Imaging in general, polarized light imaging, was used in 2 dimensions in particular, where you can learn orientation in the plane kind of perpendicular to the viewing direction. But the inclination of something that is optically anisotropic out of the plane still remained kind of a challenge, and so we are trying to solve this, and I'll give you some hints where this is going. Yeah, so to start with, just a primer, polarized light is if you have a beam of light that travels along the z-axis, then the electric field, the electromagnetic field that carries that light, that beam of light, has an amplitude and a direction. An electric field is a vector, and so it has an amplitude and a direction. In this case, let's say in a plane that is in the xy direction, and it's always, the electric field is always perpendicular to the transmission axis and to the propagation axis. And so it can be linearly polarized, so the electric field always stays in that plane, or it can be circularly polarized. And so the electric field vector within a wavelength, which is only 500 nanometers typically, rotates, and that's circularly polarized light. And then you can have anything in between. You can have elliptically polarized light. So this is called circular because the electric field, the point kind of that you connect between the different directions of the electric field is a circle when you look down the axis, the propagation axis here. Or it can be elliptically polarized. So these are different forms of polarization that all of them kind of elude us because our eye, the photoreceptors, are not sensitive to the polarization state of light. So now the next thing I had in mind were some demos, and Ariel tried hard, but it doesn't really quite work out. If you come later, if you're interested in looking at some of these props and how they affect polarization, I would ask you to come down and we can look at them together. But there are, for example, these kind of now objects, if you put strain on them. So if you put strain on them, you might have seen that in demos. I grew up in Munich, and I love to go to the Deutsche Museum in Munich, which is a technical museum, and it has all kinds of demonstration objects and so on. And so if you put strain on them, you can actually see that the strain field induces birefringence. Now, birefringence is something that converts one polarization into another, like what I showed here earlier. If you have, for example, linear polarized light like this, and you can generate linear polarized light, for example, through these kind of filters. Regular light that comes from an incandescent bulb or from a fluorescent bulb or from the sun is typically not polarized because it has this statistical property of having all kinds of polarizations mixed together. And then this is a filter that Edwin Land from Polaroid discovered in the 30s or 20s, also 1920s, that if you put iodine crystals, small needle-shaped crystals, into a plastic matrix and then stretch the matrix so that the polymers become all aligned, they also align these iodine crystals, and then they absorb linearly polarized light that is actually perpendicular to the crystal. No, they absorb the light, the linearly polarized light that is parallel to the crystal axis. And then so you have linear polarized light then that comes through this filter that is perpendicular to the needle axis of the iodine crystals. And then you have a second one. You can have a second one, and you can buy these at polarization.com. You can buy these filters, and you can align them parallel. Wow, I can even do this here, maybe. Oh. You can align them parallel, and then you cross them. So now, here they are aligned parallel, and then you cross them, and it's dark. And now if you put something in between, if I had a third hand, I could do that maybe. Then you can show that this lights up, because one polarization, the linear polarization, is turned into another polarization, and so it partially passes the second polarizer. And that's essentially what a polarized light microscope is, only that you have microscopic optics, not macroscopic optics, like in your eyes. And so we can do that later if you're interested. So these are the demos. And then this is a traditional type polarized light microscope, where you have traditional type optics, microscope optics from an arc lamp. Down here, you typically need a good amount of light. You put an interference filter in to select a wavelength, and then you have a linear polarizer and a linear analyzer that are crossed typically, and so your field is dark to start with. And then you have a compensator. That means that you uniformly across the field change the polarization slightly from linear to something elliptical, typically. And so you have a gray background here where the where you have the compensator. And then if you put a object, like in this case, it's a nice demonstration object here in the microscope. It's what's called an aster. It's an extracellular preparation where you have a protein complex called the centrosome here in the middle, and then you have these biopolymers, microtubules, radiating out in all directions. They are nucleated. Yeah. Yeah, they're nucleated at the surface of this protein structure, this spherical protein structure, and then they radiate out in all directions. And here you see the kind of the hallmark of a traditional polarized light microscope, where some of these arrays of microtubules show up dark, some show up bright, and some don't show up at all. And those are the ones that are parallel to the polarizer and the analyzer axis. So this is the orientation-dependent contrast. It still is a beautiful technique, and it's a very quantitative technique, because you measure a material property, essentially. You create contrast due to a material property, and not through labeling or so. It's an unlabeled, unstained, can be a living cell, and I'll show you some pictures later, that has not interfered with. So it's Shinya Inoue really discovered that in the 50s, that this can be a beautiful technique to study cell division, for example, what kind of structures come and go as a cell divides, and you can make beautiful movies and study the structural composition. Now, the orientation-dependent contrast always got in the way, that to study this more quantitatively, because you never quite knew what was the orientation of the. If you have filaments, it might be obvious, but in other situations where you have membranes, folded membranes, and so it might be not so obvious to know what the morphological feature is that you're actually looking at. And so when I joined at the MBL, we came up with this. We came up with this scheme that we can, instead of having this mechanical rotatable birefringent plate that's called the compensator, we used liquid crystal devices, and we built up a small. It's maybe the whole thickness of this. Let's see, the whole thickness of these three elements: a linear polarizer to start with to get the light polarized, and then two liquid crystal devices that gives you control over the polarization of light within milliseconds. You can change the polarization from one state into another uniformly across the whole field of view, and that the whole thickness here is maybe 9 millimeters. So if it's bonded together, so it's a very innocuous also for imaging. It doesn't really affect the imaging quality. This thin plate. And so it's an easy way of adding really quantitative features to the traditional polarized light microscope. The polarization analyzer here is no longer a linear analyzer. This is a fixed analyzer. It's for circular polarized light. So that this object that is this array of microtubules, now if you turn this to circular polarized light that has the opposite handedness to the one that is filtered by the analyzer, then the background looks dark, and any part of the field that has some birefringence will show up to some degree, and then you go quickly through some elliptical steps and you see kind of the symmetry is changing, even though the object is not moved in any way. And it's in a very short succession. You take these several steps, and then you can have then we have an algorithm that combines these images, and you can learn how much of a difference in phase really the light has for different polarization orientations. That's called, this difference in phase is the retardance. This differential phase is the retardance. And then you have a slow axis orientation. That is when the birefringence kind of generates in your material, generates a direction that has a high refractive index and a direction perpendicular to it that has a lower refractive index. And so the difference between the two refractive indices is called the birefringence. It's a delta n, n for refractive index. And then the slow axis is the direction for which the refractive index is highest. So the high refractive index means is slower. The speed of light is slower for that polarization. And so that's where the names come from. So this is now the output kind of from the computer. You cannot see these images in the eyepiece. This is something that's like in X-ray tomography or so, where you don't see the image directly, but you have to go through computational steps. And so these are the types of algorithms. It's very simple. It's pixel by pixel. So it's image arithmetic. And out of these 4 images, you can generate the 2 images that tell you how much anisotropy there is and in what orientation. This is another way of representing more intuitively. So yeah, the orientation image looks a little funny, but because what is it? Polarization is a tensor, or it's an orientation. It's not a direction. So 0 and 180 degree orientation is the same, and so you have that sharp step here. And if you this is a more intuitive way of indicating what is the slow axis orientation for these but you do know it for every point in the image, and then this is a color representation. These are microtubules that are now in vitro preparation, and you can kind of pin them down to the cover slip of the microscope slide that you prepare, and you can see that you're really very sensitive. This is a single microtubule. These are 2, these are 3, and here they splay out again. And each time, the measured retardance of this object, of this thin, it's 25 nanometer thin object, microns long, it steps up in a quantile way, you see that the imaging is really limited by the numerical aperture, that is by the angular range over which you collect the scattering from this. And in the end, it's a scattering problem. It's it doesn't really make sense to talk about a refractive index of an object that's 25 nanometer thick. The wavelength of light is 500 nanometer, and usually a refractive index is something that is averaged over several wavelengths. And so it's really a scattering problem that you have to think of, and that one polarization is scattered a little more with a little more phase shift than the other polarization. And so, but it's a very quantitative way of assessing. Even so, I don't resolve the 3 microtubules just by measuring that it's 0.2 nanometer retardance here. I can know that this is 3 microtubules. Up here. Let's see here. Aha. Yeah, so hold on. Here. So this is a field of cells. These are spermatocytes from a crane fly. A colleague of mine who makes these beautiful preparations, they this is one time point, and you can see that here that this is one cell, this is another cell. They are synchronized by some means, and they have built a spindle. So these are microtubules, like we saw earlier, that span between the two poles. The aster that I showed you earlier, this extracellular prep, is here. The centrosomes is the poles of the aster. They built this structure that's called a spindle, a meiotic spindle in this case. These are the two cell divisions before in this case, the sperm forms that reduces the chromosome set. As you know that the chromosomes in the sperm and in the egg are half of what it's usually, and so these are divisions, but they go on just like any other division. Most of it is the same mechanisms and everything. These are microtubules that span this place, and then the chromosomes attach to these filaments, and then at some point they will move apart, and that's what now we jump back in time. So now you can see the spindles forming, the microtubules attaching, and then once it's completed, and these are all still to a large part a mystery how this all works mechanically, from a signal point of view. When do the chromosomes know, kind of, or when does the cell know that the chromosomes are all attached properly? There has to be a checkpoint, kind of, and so all of these things are under investigation at MBL and other places. But we help there with the kind of the structural basis of these functions. And now, so now I want to come to the kind of the property that we really cannot tell yet, and we are working on revealing, and that is the third dimension. That what I showed you before is now we look from the side. So we have a side view. The microscope axis is vertical here, and then we have a focus plane that is horizontal, and that the images that I showed you earlier goes through the middle. Now, if we know from other techniques that this is a Kush ball, that this is really in this extracellular prep, we have about the same amount of microtubules going in all directions, all three directions. And if we change the focus to something through here, we see a reduced retardance, and it is because the projection of these type of microtubules, the anisotropy projection kind of in this plane into this plane is reduced. And in fact, it is zero if the microtubule bundle is parallel to the viewing direction. It's like intuitively, if you think of a microtubule as a round cylinder, and if you look at end on a cylinder, there's no anisotropy. Whereas if you look perpendicular to the cylinder axis, there is more polarizability along the cylinder axis versus perpendicular to it, and you have a high anisotropy. But if you look down, it's only it's not anisotropic. So that's why in a single viewing direction you don't really resolve, and you don't get a signal from this anisotropy that is parallel to the viewing direction. So how can we solve this? These are the perspective schematics, and so one way we kind of visualize this more clearly is with a very thin layer of calcite. Calcite is that's actually here. This is a beautiful large calcite crystal that is highly birefringent, and this layer here is only 300 nanometers. So it's a very thin layer of calcite but it is crystalline. If you had polarizers, if we had polarizers here, if you look down the obtuse angle of a calcite crystal, it is not birefringent. But if you look perpendicular to this angle or at any other angle, then it is birefringent. And so the crystal here, for example, seems to have no retardants, no birefringence, whereas others are more highly birefringent, others are kind of in between. So what's happening here? And what is what the here. So this is kind of how these layers are made. It's an amorphous calcium carbonate. Calcium carbonate is calcite is one crystalline form of calcium carbonate. And then with heat, you can make this thin layer, and then you get in the polarized light microscope. You see these. You have the retardance of these thin layers that change, and so the dark crystal had its optic axis parallel to the viewing direction, whereas the bright crystals had them perpendicular to the viewing direction or at some angle. And so how can we visualize this? And that's when I came across a technique thinking about this so these are the different orientations, and so I came about a technique that's called light field imaging, and that really intrigued me. It's a technique that long time ago at Stanford they used. For example, they had a wall of cameras, regular cameras, and the camera. This wall of cameras were all tilted in a slightly different way, so that they looked at the same scene, but because of the slight tilt in angle, they all had a different viewing angle. And so the same scene, seen from different viewing angles, that's called really a light field. If you know how much light has gone through a small volume in space, in different directions. That's a term that was, I think, in the 20s or 30s, 1930s, were coined a light field. And if you know not only the position, but also the direction in which the light propagated through a volume element. And then a graduate student of Mark Lavoie, who kind of worked on this field in computer graphics. Maybe Mark Lavoie rings a bell with some of you. He now works with Google. And he had the idea, why don't we, instead of making this large field of, this large array of cameras, we put in a single camera with a single regular macro lens, a micro lens array in front of the sensor, in front of the CCD or CMOS sensor, so that you have about, let's say, 16 by 16 pixels behind a micro lens, and the sensor is one focal length away. That means that these 16 by 16 pixels, they give you all different directions, the light. So this is what's shown here in the top. That's what's shown here in the top, on the top here. So these are the micro lenses, and then a light that comes from one point in your sample is captured by the objective and then focused onto the image plane. So this is the regular image plane where you would put the sensor, but instead you put the micro lenses there, and then behind the micro lenses, one focal length away, is the sensor. And so these 16 by 16 pixels correspond to different directions of light. And then so different directions here they're relatively, it's a low angle. In microscopy, you have a large angular range here that is converted into a small angular range and a larger picture. That's kind of optics, the reciprocity of angular and spatial resolution. And so you have these marginal rays here have a much higher inclination angle in object space than they have here in the image space. And so you sample the different orientations behind these microlens. And then you go one microlens over, one microlens over. You sample here the space still, you sample the volume still spatially in terms of position and angular. So it's a hybrid data set what you have here. And it really is tomography. You can think of it as tomography in that you have different, you know how much light has gone through and what, for example, the polarization history is of that light if it goes through the sample in different directions. So that's, and you can see that the only hardware change that is actually required here is this camera, is this light sensor. There's no other change in here. There's all the same optics. And so it seems a kind of a magic trick in order to get the kind of information from the light that you collect on the in the image plane still not used to the clicker here. So it's I like to think of it as you sort the rays differently, and there's no free lunch in a way that you do give up some lateral resolution, and especially in microscopy, you don't really like to do that. So this technique, while it has been on the available kind of since 2008 or so, hasn't really made much impact in microscopy because its focus on high resolution, on super resolution, now kind of doesn't make it but we think for polarized light, you get this additional information that how polarization fared as it went through your sample for different orientations, and that really makes a difference. That makes it possible to here in this case, for example, you see you might recognize the same field. This is now what the light field image looks like with the microlenses, so that you have so behind each microlens, for example, this one that's here circled, you have this back focal plane that is the orientation image. Behind each microlens, you can know how much retardance, if you add this polarization analysis, how much retardance have different rays at different directions have encountered. So how much retardance is imprinted on the polarization of the light as it travels in different directions through this object. And you can see that, for example, the one that is normal, that is the center of this circle, these are rays that have traveled parallel to the viewing directions, whereas these marginal rays that have had a high angle, they have picked up more, quite a bit of retardance, whereas the one that travels right down the optic axis doesn't pick up any retardance. And so you have what's also called in polarized light microscopy conoscopic imaging. Conoscopic, so is the angle in Greek. So it's a traditional technique, but here applied many times, kind of over the field of view. And we have applied this, for example, with a group at Brandeis University, and they're studying colloidal liquid crystals, like what I have worked with for a long time before I joined at MBL. These are virus particles, FD. They're all the same length. That's part of the beauty here with you can do soft matter physics with biological particles because they are all the same lengths. And so here you have these virus particles in a schematic, and they form, if you mix them with regular polymers, they get crowded out kind of and form these beautiful disks of aligned particles here. And at the edge, because they have a tendency to twist against each other, which they can't really do in the bulk, but at the edge they can twist. And you can, in fact, see that very nicely in these light field images. So that's a direct application here of this. We would like to take this, and there are, maybe you've seen a camera that's called a litro camera that can take pictures, and you download it to your computer. The picture looks like a regular camera, and then on the computer you can determine where you want to put the focus. You can change the focus of this data set, or you can change the perspective. You can you can move you can seemingly move the object, or you can view the viewing angle in the image that you create on the computer. And so you have a 3D data set, a 3D volume. And that's the same type of technique. That is this light field imaging. That's the graduate student. He founded the company Litro, which implemented this technique in a regular handheld camera. And so we'd like to move this also to this 3D type viewing, reconstructing 3D objects, including their anisotropy, their polarization anisotropy, their birefringence axis, not only in the plane perpendicular to a single viewing direction, but also how much it is tilted. So it's 3D orientation. It's something, as you can see, the hardware is really simple, and really the magic is in the software that reconstructs these images, and that's something we are working on. We aren't really there yet. We have a graduate student who has, I think, got a handle on fluorescence imaging. So in fluorescence imaging, you also have dipoles that emit the that emit the light, and the light is polarized of a dipole, and so you can also learn orientation of dipoles when they're attached to certain structures, and that's something we are working on right now. We will come to birefringence to do the same in birefringence, to generate an object, a reconstructed object that also tells you, for example, the axis of the spindle, and it makes contrast of spindles that are not just in the plane perpendicular to the viewing direction, but also tilted to it. So this is kind of our goal. We do have we support people who would want to implement this technique. We call it now the optical. The company that used to pick up the technology, Cambridge Research and Instrumentation, was a beautiful small company that we worked with, and they have licensed some of the patents that we have on it. They were bought up by PerkinElmer in two steps. The first was Caliber. Caliber bought up CRI, and then Caliber was bought up by PerkinElmer, and it became very difficult to work with them. And in fact, they have dropped the product, but I think people are still interested in this technology, and so we have created this OpenPolScope resource at MBL, and And so we are in principle ready to support anybody with this technology. So polarized light reveals the degree and orientation of molecular order simultaneously for the whole field of view. And then the single view microscopes reveal the 2D orientation, and then the multi-view microscopy and light field microscopy is one of those multi-view imaging. We have another setup at MBL for fluorescence imaging, really only that has simply two microscope chains through imaging setups that are at 90 degrees to each other. And they have the additional benefit that you have isotropic resolution. In a regular light microscope, you don't have isotropic resolution. You have good resolution, let's say, in the plane perpendicular to the optic axis. But along the axis, when you focus up and down, you actually have about for high NA objects, you have about a factor 2 less resolution. And then for low NA, for kind of macroscopic type imaging, where you don't have that large of an angle, viewing angle, over which you collect the light, you have maybe 10 times less resolution. So whereas if you have two imaging systems that look along perpendicular axis to each other, you have isotropic resolution. Kind of one view gives you the high resolution in the direction the other view lacks the high resolution. So there are different ways of implementing multi-view imaging, and we are using both types of imaging. Yeah, and then I think the acknowledgement really Shinya Inoue, who brought me into this field. I'm really grateful. Charlene Mehta, he is now with Chan Zuckerberg BioHub in San Francisco. He's there, the leader. He worked with us for a couple of years. Mai Tran, Amitabh Verma, and Michael Schriebach, who all at MBL work on this. And then the University of Chicago, Patrick Lariviere, he's a professor in radiometry and this inverse problems in imaging. And Talon Chandler, a very gifted theoretical physicist who does helps us in this inverse problem of polarized light field imaging. Thank you.
We have some time for questions.
Ariel. Yes, so I've also read about your, besides the multi-view, the multifocal
yeah.
That you have
right.
published. Can you tell us a bit about that?
So Sarah Abramson has implemented that. She has worked with Mats Gustafsson, who really first implemented this way of generating several focal planes from a single setup, from a single focus setup in your microscope. And what it is it's a regular microscope setup, just like I showed you, but in the back focal plane of the objective. That's the Fourier plane kind of imaging. You can put a diffractive grating that is designed specifically to generate different focal planes in the image plane, in your regular image plane. So you have kind of an array, and it all depends on how you construct this diffraction grating. You have several different focal planes, as if you had kind of sequentially focused your object into different planes. It only works with a relatively monochromatic light. Of course, you can't use, because it's a diffractive type effect. And it doesn't generate different viewing directions. So all these focal planes are as if you had really focused your specimen differently. but they have not changed the viewing direction. So for our case, in order to get the tilt angle of the optic axis, it's not really suitable.
And the distance between those focal planes, how small can it be, or large?
Yeah.
In practical terms, you know, when you create this grating.
Yeah. I mean, I think you would want to set it, design it so that it samples again the resolution. Let's say the resolution is 200 nanometer laterally, and that is kind of a high NA oil immersion optics. And so you want to sample that on your detector with the Nyquist limit of at least 2 pixels within this 200 nanometer range. Axially, because you have a larger spacing, and you can do that with designing this diffraction optics accordingly, you would want to probably have 2 planes within the resolution limit of 500 nanometer. And I think you can do that depending on how you design the optics, the diffraction grating. Yeah, so that's a way. You can do 2 by 2 or 3 by 3 or 5 by 5 different. You always, depending on the camera you have, you might limit your field of view that way because the camera can only take that many images.
Any other questions?
Mm-hmm.
Patrick?
So you showed some very low birefringence samples measured, and I was wondering what, in like a practical setup, limits that sensitivity.
Yeah,
kind of what's
that's a good.
What's the lowest
Right.
you've seen measured?
So in the end effect, in the end, it's the shot noise. It's the natural variation in intensity that you get from just counting a finite number of photons. In most cameras, the scientific CMOS cameras or so are really shot noise limited in there. So if you count 10,000 photons, you have 100 plus minus, and it is this different. It is the difference between these elliptical. You remember there is one circular setting which is good for kind of knowing the baseline, and the dark frame, and then the elliptical settings carry much of the information on how much anisotropy there is and in what orientation. And it's the differences between these 4 elliptical settings that determine that. And so if you, for example, if you take if you have a clear sample like the ones that Peter Kazansky is making, and you put this in the background, and you have no birefringence in there, and you take 2 successive images of that, you will measure some retardance and that will change with every image taking will change because there are small differences due to the shot noise, and that's kind of what sets your limit. And that if you count long enough, if you expose long enough, and if your camera can take 1,000,000 photons per pixel, you can have very low shot noise, and so you can go to 0.01 nanometer or maybe even lower in retardance.
Interesting. Tom?
How much do we know about the mechanisms that allow these living creatures to detect polarization, and can we emulate those in our detectors?
Right. So that's part of a group, a behavioral group at MBL, who is exploring that, how they detect it, why they detect it, kind of how do they change their behavior. And in some cases, it's to detect prey, because the light, especially the light that kind of travels at a specific angle from the surface of the water is pretty highly polarized. Whereas our light, the skylight actually is polarized. If you, and I actually want to offer, I have little polarizers here, and so you can have a polarizer and you can explore different things. Oh, this is, for example, I'm not getting to your question. You can tell that the rainbow is polarized, and you can think about it why. But what makes them polarized? There's some. Whenever light interacts with matter, it typically either gets polarized partially. It's of course in most cases it's partial polarization. That there's a small component that is polarized. And so, yeah, polarization gives you clues about the environment, and one of them is the prey, because the light in water gets polarized when it travels from air into water. And then what it causes, I don't know, to be honest, right now. It's I do know. And that is in the human eye, the retina and the rods and cones, they have membranes that are disc shaped. The membranes are in the membrane is the chromophore that absorbs the light and then converts it into a signal that is transmitted by the nerve cells. And the chromophore has its dipole moment in the membrane, and so the membrane can the chromophore can rotate in the membrane. So there is no preferred orientation of the chromophore dipole for these disc-shaped invaginations of the rods and cones. They have these discs that stack on top of one another and make this absorption efficient for a single rod and cone. Whereas in some fish, and I think they're similar in insects. Many insects have polarized light sensitivity. These are not discs, but these are rods. And there you have a remaining orientation preference. If these membrane rods, so they are cylinders, let's say, and there you have a remaining preference in orientation if you average overall cylindrical positions, and that makes it polarization sensitive in fish, for example, anchovy and in other fish eyes. They have this different geometry in the rods and so. Mm-hmm.
So you mentioned with the field camera that you take a hit on spatial resolution, which makes sense.
Yeah.
In fluorescence microscopy, there's various kind of statistical analysis techniques to recover spatial resolution at the cost of, say, temporal resolution. Is there an equivalent in kind of polarization microscopy?
So by you're thinking of changing the perspective or so, and as we move, we can accumulate that type of information to improve resolution.
Yeah. Or just from the sample itself, if there's a time-dependent change.
Yeah, in a way, the sequential settings that we go through kind of mimics that type of resolution where you average over time and kind of get different information and combine that in polarized light imaging. Yeah, it's interesting. There is, for example, there is an effort right now in the type of imaging that I showed you, this polarized light, and also phase contrast, for example, or differential interference contrast, if some of you know this they give you really structural information. They don't give you molecular information in terms of what kind of molecules. We can't tell that it is microtubules, that the chemical nature of these biofilaments that form the spindle, that form that make the birefringence, that this is actually this tubulin, the chemically tubulin. The polarized light and it can't give you that. Fluorescence imaging gives you molecular specificity because these labels only bind to actin. And if you see filaments that light up in fluorescence imaging, this must be actin. This must be microtubules. This must be a membrane protein or so, because it's molecularly specific, the binding. There is an effort now to kind of combine these fluorescence imaging and train, it's a neural network thing, and train a neural network to identify certain features that we don't perceive right away, that is specific to actin in a DIC image, in a polarized light image, that is specific. The morphology is just a little different from microtubule. So to train a network to identify molecular specificity, to add that to what you can know from these molecularly unspecific techniques.
Great. I think for reasons of time, we'll wrap up there. Rudolph's around through, including lunch. So if you'd like to have a more detailed discussion, please do come down and chat with Tom, who's, I think, organising the schedule. Let's show our appreciation again for a very interesting lecture.
Thank you.